Good morning. I was just trying to get my tweet out, and I have failed. So we're failing in our promotion so far this morning. Margaret mentioned her ties to Boston. Dr. Glimcher, you are the quintessential hometown girl.
I am.
Good. You
I am.
went away to Cornell a while and are now back here at Dana-Farber. And I wonder, you've just come back just before Christmas, just before Thanksgiving, I think you
October.
just came back.
Yep.
Yeah. And it strikes me that you were supposed to have a honeymoon period in your new role.
What's that?
But you seem to have come into a real maelstrom of time in biotech, in medicine, and of course in the political environment that we're in today. And I wonder if you would just start giving us sort of that 30,000-foot view of what's happening, particularly in this town today, in terms of where it's bringing advances in medicine and technology, bio, everything.
Well, I think that Boston is unparalleled, actually, as a town for all of those things. We have been in the forefront for years in biotechnology, in new discoveries, and the Boston community is just an intellectual dream, right? This is why biotech and pharma companies want to be here, is because Boston is the place to be. The intellectual capital here is just amazing. As you said, I was born and bred in Boston and went through the Harvard system for my whole career. My dad was chair of orthopedics at MGH and Children's, and now my son is a cardiothoracic surgeon at Mass General. So we've got a long family history here, and I managed to stay away for 4.5 years, being dean at Weill Cornell, which was fun. I loved it, but very much wanted to come home because I think Boston's an incredibly special place. And it's an incredibly special place right now for cancer, as well as many other research areas. I think I was particularly happy to come to the Dana-Farber. It is an amazing institution. The quality of the research faculty and the quality of clinical care that Dana-Farber delivers to our cancer patients is something I have never seen anywhere else, and I keep on hearing that theme over and over again from everybody I talk to, and it is really true. So I was delighted to come back here, and particularly because this is a cancer institute. I'm an immunologist by training, and as you know, there have been really 2 revolutions in cancer that have moved the needle in a way that hasn't been possible for 100 years. So the last 10 years have seen an amazing revolution in cancer research and cancer care. The first revolution I think of as being precision medicine, which I know is a subject that's of great interest to everybody. And that was the recognition, and this was really jump-started at Dana-Farber Cancer Institute, the recognition that each patient's tumor has a unique genetic mutation that drives that cancer.
It's a signature,
It's a signature. So what makes a normal cell turn into a cancer cell? Something goes awry in that cell, and what goes awry is a change in the DNA in that cell, so that it becomes activated and starts to proliferate and grow. The question is, could we identify those altered genes in the tumor cell? And so that was the precision medicine revolution, which, as you know, President Obama announced at his State of the Union address, and that's, although it applies to other diseases, it has been most often used in cancer. And so Dana-Farber and other cancer institutes set off to begin to identify these genetic mutations and then to devise drugs that specifically target them. I'll give you an example of a drug that does that. Gleevec is a drug that treats chronic myelogenous leukemia and has made that disease essentially a chronic disease. I will hesitate to use the word cure, but it has made chronic myelogenous leukemia into a chronic disease. People can live with it, and that's because this particular drug, Gleevec, targets that particular bad actor gene in the tumor cells.
So this is so remarkable, and people have also called this personalized medicine, targeted in a way. And there's so many things to ask you about in this, but I'm struck by this sense that I think as a layman we look at it and say, well, if I could just give you, what, my DNA, something, you could see it, you could, boom, right? Instant. Now everybody has the expectation perhaps that, well, if you can figure that out, you can cure anything instantly.
Well, there's a big gap between figuring out what the driver of cancer is and designing new drugs to combat it. I would say at Dana-Farber, we're the only cancer institute in the country that sequences the DNA of every single cancer patient that crosses our threshold. We've sequenced 16,000 people's DNA of their tumors to identify those mutations, and actually about 60% of those patients, knowledge of that mutation has led to clinically actionable treatment. So we can say, all right, you have this mutation, you should either be on this drug or you should enroll in this clinical trial that's testing a new drug if a FDA-approved drug is not yet available. So I think it has been absolutely revolutionary, but we're reaching a time when we're recognizing that one drug isn't enough, because the tumor, unfortunately, is very smart and it outwits us. And once you've attacked that one gene, another gene pops up to drive the tumor, so it escapes. The tumor can circumvent the drug. And so we realize that the situation now is quite similar, actually, to where HIV/AIDS was years ago. As you know, HIV/AIDS used to be a lethal disease, but now a 20-year-old that becomes infected with HIV has virtually a normal lifespan, and that's because, thanks to wonderful basic research in many laboratories, it was discovered how to attack the HIV virus. And you had to do it by using a combination of drugs. And using that combination of drugs has turned HIV from a lethal disease into a chronic disease. Now, cancer is a little bit more complicated because there are many different kinds of cancer, many different mutations. But I think ultimately the future of cancer treatment is going to be using a combination of drugs, both those that target those precision medicine targets, but also the new revolution, which is activating your own immune system.
Speak to us about this. You are an immunologist in your training and experience. This is really the next area of breakthrough, I guess.
It really is, and there are some transformative medicines out there right now that have led to durable remissions in diseases that were previously lethal. The idea is that we have an immune system, right? We have this immune system sitting within us that is dedicated to fighting off foreign pathogens. Those can be viruses, bacteria, and they can also be tumor cells, because tumor cells don't look like our own normal cells. They display surface proteins that should be recognized by your immune system as, oh no, that doesn't look like us, we're going to kill you off. And 99% of the time that works beautifully. We probably all have some rogue cancer cells floating around, but our immune systems just get all excited and knock off those tumor cells. Doesn't work all the time. And so the question for scientists for 100 years now has been, how can we further activate our immune system to kill off the tumor? Because that's what it's supposed to be doing. But some of those immune cells that should be doing that are, quote, exhausted, and they're just not active enough to kill off the tumor. 100 years ago, there was a surgeon in New York City named William Coley, and he's probably the father of immunotherapy, because he noticed that when he removed tumors from some of his cancer patients and the wound got infected, sometimes that tumor didn't come back. And so he reasoned that there must be some system inside the body that was, while it was reacting to the bacteria or the virus, also killed off the tumor cell. And that turned into Coley's toxins, which was a bottle of bacterial mixture
Which was given to
which was given to patients, and had some effect. It was supplanted by chemotherapy and radiotherapy eventually, but it gave a hint. And it's taken us 100 years to actually make good on that initial discovery. And that was done by realizing that you could activate the key immune cell by treating it with a protein therapeutic, which is an antibody, that binds to that T cell and activates it. And that resulted in several of the drugs that are on the market today. Those were first tried in melanoma, and they were tried in patients where the melanoma had spread to many organs, so-called stage 4 metastatic melanoma, and those patients were really at death's door. And for some of those patients, treatment with these new therapeutics has resulted in what seems to be a durable remission that can last as long as 10 years, in fact, still counting. So that was incredibly exciting. That was an incredibly exciting discovery, and I must say the first clinical trial in patients with these new drugs was done at the Dana-Farber Cancer Institute by one of our physician scientists, Dr. Steve Hody, and published in the New England Journal of Medicine years ago. So it sounds like, okay, you've done it, you've cured everybody, but in fact, it's just the tip of the iceberg, because only patients with certain tumors respond to these immunotherapeutics, and there are some cancers that really don't respond at all to them. And so we have a lot left to do.
And so what is the key to being able to do that? It seems that collaboration becomes increasingly important in this environment where you've made advances in these transformative treatments, but what do we need here? You have talked about your concern about funding for basic science and everything else.
Well, I think there are a lot of things we need. We need more funding for basic science, because if you don't do basic discovery, then you don't have anything left to translate into translational medicine, which then leads to drug development and new cures for patients. So the NIH budget is not sufficient in its current form to fund all the important research that needs to be done to get underneath the tip of that iceberg and figure out why some patients respond and why some patients don't respond, why some tumors are sensitive to immunotherapy, but others, like pancreatic cancer, ovarian cancer, prostate cancer, breast cancer, are not very susceptible to immunotherapy. There's a lot of basic work that needs to be done, and the bottom line is inadequate funding from the government.
And do you anticipate, you came from an administration, we came from an administration which launched its moonshot toward curing cancer. Now we're entering a new administration that has clearly different goals in a lot of arenas already. What is your concern for that?
Well, I was honored to be on Vice President Biden's Blue Ribbon panel to determine what themes and what projects would be targeted by the moonshot. And there are a number of important ones, but to your point about collaboration, one of the key themes of that Blue Ribbon panel was collaboration, share data, because if we work together, then we're going to move the field faster. And so Dana-Farber, again, has been a leader in that by creating, with another cancer center, a program called Genie program, the Genie program, whereby I think 7 or 8 institutions have banded together to share all their data as they sequence patients' DNA. They're going to share all the data, and then in 6 months, those data are made public and widely available. And the Vice President, I have to say, really stressed that during our meetings, that you've got to get together and share data. And I totally agree with him, and Dana-Farber certainly is on the leading edge of doing that.
So the 16,000, I think you said, that you have sequenced.
Those data are shared. They are shared with those other institutions, and some of it now has been made public. And we will continue to do that, and we will continue to sequence more patients, and we'll sequence not only the tumor itself, but the immune system that surrounds that tumor. If you think of a tumor as a castle, and the problems, the cells that block the activated lymphocytes from getting in there and killing the tumor like a moat, you got to get over that moat. And so it's really important to figure out why parts of that immune system are blocking the key cells from getting in and killing the tumor. So we're going to add that to our sequencing. Now, this is not reimbursed. This kind of expensive test is not reimbursed, and so we've been able to support it initially with philanthropy, but it gets increasingly more expensive. And
so that is that other element of collaboration.
That's right, because the more patients you can sequence, whose tumors you can sequence, the more information you're going to have, and the more understanding you're going to have of why some patients respond and some patients don't. I do have concerns about the new administration in the sense that the $4.8 billion that was put aside for a number of programs, including Cancer Moonshot, Precision Medicine, Opioid Addiction, the Brain, is guaranteed for 2017, fiscal year 2017, but has to be approved every year after that by Congress. So the Moonshot funds, for example, are spread out over 7 years. So this year, we're allotted roughly a little over $300 million to distribute, the government is, but it has to be approved in subsequent years. And, I hope the Congress will continue to approve these allotments. They're absolutely critical to research into cancer, to treating opioid substance abuse, and many other things. But we don't know that. We don't have any surety for that. And of course, I'm also concerned about the ability to treat all of our patients. The repeal and replace the Affordable Care Act is in progress. There have been some recent statements about it. A 19-page document was just released. And it appears that Medicaid has been targeted. We treat every patient that comes across our threshold. And yes, we, like other hospitals, lose money on treating Medicaid patients, but we would never deny care to any person that walks across the threshold of Dana-Farber. And it looks as if there's going to be downward pressure on Medicaid reimbursement. We have very generous Medicaid reimbursement in Massachusetts, but 1 out of every $4 that we get from Medicaid comes from the federal government, and it looks like that might be under threat. Talking about Medicaid block grants to the states, I'm concerned about the 22 million people who now have insurance. The individual mandate will no longer be required, according to what I have read most recently.
But even something like pre-existing conditions could, in fact, affect, and this was sort of a leap to me in reading about this, but okay, if I've got a pre-existing condition and I no longer get coverage, then I don't want to reveal my data, which then becomes a problem.
That's true, and also you may not get care because, again, the administration is talking about forming a high-risk pool. And I'm not sure how that's going to play out. But right now, as you know, you absolutely, no matter how many risks you have, how many diseases you have, how many chronic diseases, you get coverage right now under the ACA. So that is another area of concern, Joie.
Yeah, we do have a microphone out here in case anybody has questions. I do see that there's a great deal of attention to what you're saying and the concerns here. To go back to where we started to talk about Boston, I mean, of course, this weekend we saw scientists in the streets, and for those of you who were part of that protest, I have to give you some props for the signage. Very interesting signs come from scientist protesters. But I think, this is, it does seem that scientists are now forced into the political arena, whether they want to be or not, forced to come forward with their views on the political environment, when I'm sure what you would rather be doing is taking care of patients and
Well, I think that scientists have always been involved in appealing to the government, whether it's the state government or the federal government, for additional funds for research. I think that the necessity and the value of biomedical research is not widely appreciated, and I think we have to take some of that blame ourselves as physician scientists, because we're not getting out enough and talking to the lay public about why it's so important to do biomedical research. And I'll give you an example of that. I mean, how many of you know that 1 out of 3 people over the age of 85 is going to develop Alzheimer's disease? Right now, we're spending $200 billion a year just treating patients with Alzheimer's disease. Well, given the age,
treating them.
Just not even treating them, because we have nothing to treat them with, right? Taking care of them.
Right.
By 2050, that number, given the aging demographics in our country and around the world, that number is going to go to well over $1 trillion. And that's going to take down our healthcare system. So the only answer to me is we got to be doing basic research to figure out why people develop Alzheimer's disease and to come up with treatments for it, or even come up with treatments that will delay it for 5 years. This is an epidemic. This is a crisis in American medicine, and I'm not sure that we have made that clear to the public.
That they understand that, live with that. When we talk about, to go back to that moonshot initiative, that aim high to cure for cancer, and you referenced this earlier.
I really hesitate to use the word cure. I mean, that was used 40 years ago, 45, 50 years ago with Nixon's war on cancer. I would be delighted to see cancer become a chronic disease. Maybe in some instances we could cure it, but when somebody presents with stage 4 widely spread cancer, I think we would all be grateful to know that we could live with that, just like people live with diabetes, or live
HIV.
with kidney disease, or live with HIV. Turning cancer into a chronic disease would be a huge triumph for us, for the world.
Going forward, you mentioned both this need for basic science funding and the need for collaboration. That collaboration has to reach, in your view, beyond what money you can count on from the federal government. That support must come, and because we've also talked about the need for drug research, the need for technology, this
is critical. I think that the marriage of academic scientists with scientists in the private sector, in pharmaceutical companies or in biotech companies, is really vital. It's a marriage made in heaven for our patients, because we want to get the discoveries that we make in the laboratory translated into new medicines for patients as soon as we can. And frankly, in academia and places like Dana-Farber, we do discovery really, really well. We can discover the new proteins that are involved in cancer. And pharmaceutical companies tend not to focus on discovery, they tend to focus on late-stage development. So they can take a compound, a potential therapeutic that isn't ready for human use, and convert it into one that is ready for human use, and that they can test in large clinical trials. So we have complementary expertise, and getting together is what's best for our patients. Now, that being said, one of the things about Dana-Farber that attracted me so much was that we've actually set up this whole pathway in-house, where we have hired a number of chemists, and have managed actually to go right from the bench to the bedside, right within the walls of Dana-Farber, but that's very unusual. We still have many, many interactions and collaborations with pharmaceutical and biotech companies, and I think that's really important, and Boston is a great place to do that.
which brings us back to how this day is going to go. The conversations, we appreciate you talking about that and setting up the conversations, because we are going to talk further about financing through the course of this morning.
Right.
Thank you very much, Dr.
Thank you.